Microbial genomics, the comprehensive study of the genetic material of microorganisms, has revolutionized our understanding of the microbial world and its profound influence on all aspects of life. This field, which encompasses bacteria, archaea, viruses, and unicellular eukaryotes, such as fungi and protists, sits at the intersection of biology, technology, and informatics. The journey of microbial genomics is a testament to human curiosity and ingenuity, unfolding from the earliest days of microscopic observation to the present era of high-throughput sequencing and big data analytics. The roots of microbial genomics can be traced back to the 17th century, when Antony van Leeuwenhoek first glimpsed the animalcules in a drop of pond water using his handcrafted microscopes. For centuries, the study of microbes was limited to what could be observed under a lens or inferred from their effects on humans and the environment. The development of classical microbiology in the 19th and early 20th centuries brought about the isolation, cultivation, and characterization of microbial species, laying the groundwork for modern genetics. The discovery of DNA as the hereditary material and the elucidation of its structure in the mid-20th century marked a turning point, enabling scientists to probe the very code of life. The birth of genomics as a discipline came with the advent of DNA sequencing technologies. The Sanger method, introduced in the 1970s, allowed for the determination of nucleotide sequences, albeit laboriously and on a small scale. In 1995, the complete genome sequence of Haemophilus influenzae became the first of any free-living organism to be published, heralding the dawn of microbial genomics. This achievement was quickly followed by the sequencing of other bacterial genomes, such as Escherichia coli and Mycoplasma genitalium, each unveiling a new layer of complexity in microbial life. With the rapid advancement of sequencing technologies, particularly the development of next-generation sequencing (NGS) platforms, the pace of discovery in microbial genomics accelerated dramatically. NGS enabled the parallel sequencing of millions of DNA fragments, reducing costs and time while exponentially increasing throughput. This technological leap facilitated large-scale projects, including the Human Microbiome Project and the Earth Microbiome Project, which sought to catalog the vast diversity of microbial life inhabiting humans and the planet. One of the most striking revelations from these endeavors is the staggering diversity and ubiquity of microbes. Microbial genomics has shown that microorganisms inhabit every conceivable niche on Earth, from the deepest ocean trenches to the highest mountain peaks, from boiling hot springs to the frozen tundra. Many of these environments are home to extremophiles, organisms that thrive under conditions previously thought inhospitable to life. By sequencing the genomes of extremophiles, researchers have uncovered novel genes and metabolic pathways that confer resistance to heat, cold, radiation, salinity, and acidity, expanding our understanding of the limits of life. Beyond cataloging diversity, microbial genomics has provided insights into the evolutionary relationships among microorganisms. Comparative genomics, which involves the analysis of genome sequences from multiple species, has revealed the dynamic nature of microbial genomes. Horizontal gene transfer, the movement of genetic material between unrelated organisms, plays a significant role in microbial evolution, facilitating the rapid acquisition of new traits such as antibiotic resistance or metabolic versatility. This fluid exchange of genes challenges traditional notions of species boundaries and underscores the adaptability of microbial populations. The implications of microbial genomics extend far beyond academic curiosity. In medicine, the ability to rapidly sequence and analyze microbial genomes has transformed the diagnosis, treatment, and surveillance of infectious diseases. Whole genome sequencing, WGS, of pathogens enables precise identification of strains, tracking of outbreaks, and detection of antibiotic resistance genes. For example, during the COVID-19 pandemic, genomic surveillance of SARS-CoV-2 was instrumental in monitoring the emergence and spread of new variants, informing public health responses and vaccine development. In addition to infectious disease management, microbial genomics has illuminated the complex interplay between humans and their resident microbiota. The human body is host to trillions of microbes, collectively known as the microbiome, which outnumber human cells and contribute to a myriad of physiological processes. Genomic studies of the gut microbiome, for instance, have linked microbial composition and function to digestion, immune modulation, and even mental health. Dysbiosis, an imbalance in the microbial community, has been associated with conditions ranging from inflammatory bowel disease to obesity and depression. By unraveling the genetic blueprints of these communities, researchers are exploring novel therapeutic strategies, such as probiotics, prebiotics, and fecal microbiota transplantation. Agriculture and environmental science have also reaped the benefits of microbial genomics. Soil and plant-associated microbes play crucial roles in nutrient cycling, plant growth promotion, and disease suppression. Metagenomics, the direct sequencing of genetic material from environmental samples, has uncovered a wealth of previously unknown organisms and genes, many of which hold promise for biotechnological applications. For example, enzymes derived from soil microbes are used in industrial processes such as biofuel production, waste degradation, and the synthesis of pharmaceuticals. In agriculture, understanding the genomics of plant pathogens and beneficial microbes informs strategies for sustainable crop protection and soil health management. Industrial biotechnology has harnessed the power of microbial genomics to engineer microorganisms for the production of valuable compounds. Synthetic biology, a field that combines engineering principles with biology, relies heavily on genomic information to design and construct microbial strains capable of producing biofuels, chemicals, and therapeutics. By editing microbial genomes using tools such as CRISPR-Cas systems, scientists can optimize metabolic pathways, enhance yields, and reduce the environmental impact of production processes. The exploration of microbial genomics has not been without its challenges. The sheer volume and complexity of genomic data generated by modern sequencing platforms necessitate robust computational tools and bioinformatics expertise. Assembling and annotating genomes, particularly from complex communities or uncultured organisms, remains a formidable task. Advances in machine learning and artificial intelligence are increasingly being leveraged to analyze genomic datasets, predict gene function, and model microbial interactions. Another challenge lies in the interpretation and contextualization of genomic information. The presence of a gene does not always equate to its expression or functional relevance within a given environment. Integrating genomic data with transcriptomic, proteomic, and metabolomic analyses, a systems biology approach, offers a more holistic understanding of microbial physiology and ecology. Such multi-omic studies are shedding light on how microbes respond to environmental cues, interact with hosts, and contribute to ecosystem functioning. Ethical, legal, and social considerations also accompany the rise of microbial genomics. The ability to sequence and manipulate microbial genomes raises questions about biosafety, biosecurity, and the potential misuse of genetic information. The release of genetically modified organisms into the environment, for instance, must be carefully regulated to prevent unintended ecological consequences. In the medical realm, the use of genomic data for personalized therapies or epidemiological surveillance must balance public health benefits with individual privacy and consent. Despite these challenges, the future of microbial genomics is bright and full of promise. Emerging technologies such as single-cell genomics, long-read sequencing, and spatial transcriptomics are pushing the boundaries of what is possible. Single-cell genomics allows researchers to probe the genetic makeup of individual cells within a community, revealing heterogeneity and rare functions that bulk analyses might miss. Long-read sequencing platforms, such as those developed by Oxford Nanopore and Pacific Biosciences, generate contiguous sequences that facilitate the assembly of complex genomes and the detection of structural variations. Spatial transcriptomics, which maps gene expression within the physical context of tissues or microbial communities, offers unprecedented insights into the organization and function of microbial ecosystems. These advances, coupled with decreasing costs and increasing accessibility of sequencing technologies, are democratizing microbial genomics and enabling researchers around the world to participate in discovery. The integration of microbial genomics with other disciplines is yielding transformative applications. In precision medicine, the characterization of patient-specific microbiomes is informing tailored interventions to restore health or prevent disease. In agriculture, genomic selection and engineering of beneficial microbes are enhancing crop resilience and productivity in the face of climate change. In environmental monitoring, metagenomic sequencing is being used to detect pollutants, track biogeochemical cycles, and assess the impact of human activities on natural ecosystems. Astrobiology, the search for life beyond Earth, is another frontier where microbial genomics plays a pivotal role. By studying the genomes of extremophiles and reconstructing the evolutionary history of life on our planet, scientists are developing models for the potential existence and detection of life elsewhere in the universe. The discovery of extraterrestrial microbes, should it occur, would have profound implications for our understanding of biology and our place in the cosmos. Education and public engagement are essential components of the microbial genomics enterprise. As the field advances, efforts to communicate its significance and implications to diverse audiences, including students, policymakers, and the general public, are crucial. Initiatives such as citizen science projects, open access databases, and interactive exhibits are fostering a greater appreciation for the microbial world and its relevance to everyday life. The ongoing expansion of microbial genomics is also reshaping our philosophical perspectives on life, individuality, and interconnectedness. The realization that humans are superorganisms, composites of human and microbial cells, challenges the notion of the self as a purely human entity. The intricate networks of gene exchange, cooperation, and competition among microbes mirror broader themes of complexity and emergence in nature. Looking ahead, the field of microbial genomics is poised to address some of the most pressing challenges facing humanity. Antimicrobial resistance, a growing threat to global health, can be better understood and combated through genomic surveillance and the discovery of novel antibiotics from unexplored microbial reservoirs. Food security, threatened by population growth and environmental change, can be bolstered by harnessing the genetic potential of microbes to enhance crop nutrition and resilience. Climate change mitigation may benefit from microbial strategies for carbon sequestration, methane oxidation, and bioremediation. International collaboration and data sharing will be key to realizing the full potential of microbial genomics. The complexity and scale of microbial diversity transcend national borders, necessitating coordinated efforts to build comprehensive reference databases, standardize methodologies, and ensure equitable access to resources and benefits. The principles of open science and responsible research conduct must guide the stewardship of genomic data and the translation of discoveries into societal good. In conclusion, microbial genomics stands as one of the most dynamic and impactful fields in contemporary science. It has transformed our understanding of the microbial world, revealed the hidden diversity and adaptability of life, and opened new avenues for innovation across medicine, agriculture, industry, and environmental stewardship. The challenges it faces are matched by the opportunities it presents to improve health, sustain the planet, and expand the horizons of knowledge. As we continue to explore the genetic landscapes of the smallest inhabitants of our world, we are reminded of the profound interconnectedness of all life and the boundless possibilities that lie within the microbial genome.